Okay everyone, this lecture is on anticoagulation and it's going to be my final recorded lecture for the blood system block. So let's get started. Now we're going to revisit the concept of homeostasis again because you have learned from your earlier recordings that the coagulation system can only work smoothly if there's a good and tight balance between clot formation and clot inhibition. If there's any disruption in this balance, coagulopathies may happen. Now you also learned about the various factors that has contributed or promoted coagulation. In this lecture, we are going to switch gear a little bit as we move on to the anticoagulation system. Essentially, the anticoagulation system serves two main purposes. The first is to prevent aberrant clot formation, because we want to make sure that clot only forms at the right site and at the right time. If clots are allowed to be formed anywhere and everywhere, thrombosis may happen, and that may result in the narrowing of our blood vessel. And you can imagine that is going to be catastrophic. Now, secondly, the anticoagulation system is also there to remove the clots once it is no longer needed, also known as fibrinolysis. And this is an important process because you wouldn't want the clots to be dislodged once the wound has fully recovered, because the dislodged clots, if it's allowed to float around in the circulation, it may end up being lodged at other tissues or organs, and that may cause embolism. So how does the body exactly execute this is what we are going to learn in this lecture itself. In other words, these are the learning outcomes that you are expected to learn by the end of this lecture. The first is to explain the physiological mechanisms of anticoagulation. Of course, the second will be the physiological basis of the fibrinolytic pathway. So one is on anticoagulation, the other one is on fibrinolysis. So once you have learned the medical sciences behind anticoagulation, that's when we move on to look at the potential disorders that may happen if there's defects in the pro and anticoagulation pathway, in particularly thrombotic disorders. And lastly, we'll try to apply what you have learned in this lecture, as well as the clinical tests that are involved to assess the coagulation system. And as usual, the first two learning outcomes will be covered in this recorded lecture, and the subsequent two will be something that we will cover and learn during the in-person discussion itself. Okay, so from your earlier recordings, you have remembered that hemostasis comprises 3+1 steps. For anti-hemostasis, there are actually 3 steps. The first step would be your plaque prevention or your inhibition of primary hemostasis. Second step would be clot prevention, and that's the inhibition of secondary hemostasis. And the third would be clot removal or fibrinolysis. And these 3 steps are exactly the ones that we will be learning today. And we're going to get started with the prevention of aberrant plaque and clot formation. And that one important factor that is critical for the prevention of aberrant plaque and clot formation will be your intact and smooth endothelial cells. And that's exactly what you have learned in your earlier lecture as well, because we say that the intact endothelial cells is absolutely necessary to make sure that plaque formation doesn't happen anywhere else but at the site of the damaged vessel, because intact endothelial cells are able to release nitric oxide and prostacyclins that serve to inhibit platelet activation and aggregation. Right? So these restrict the plaque to happen or occur only at the damaged wall. But that's not all, because the intact endothelial cells are also critical to inhibit aberrant clot formation. How does it do it? Through the release of TFPI or tissue factor pathway inhibitor. So what is TFPI here? Now, TFPIs are essentially proteins that inhibits your tissue factor. And if you recall from your coagulation cascade, the tissue factors are there to activate your factor 7. And that is essential for the activation of the extrinsic pathway. So with the release of TFPI by your intact endothelial cells, TFPI will be able to bind and inhibit your extrinsic pathway. And that will make sure that clot doesn't happen when there's intact endothelial cells. Now, other than TFPI release, the intact endothelial cells are so critical to inhibit clot formation through its surface receptors. So there are two surface receptors that are particularly important when it comes to the inhibition of clot formation. These are the thrombomodulins as well as the protein C receptors. Right? Because thrombomodulins and protein C receptors are there, as the name suggests, to inhibit or activate thrombin. Right? That is the function of your thrombomodulin. And your protein C receptors are there to activate your protein C and S. And the protein C and S is especially important in the anticoagulation system. And that is something that we will learn in your next slide. Alright, so how does the protein C and S work in inhibiting aberrant clot formation? And this is something that we'll cover in this slide. Now you can imagine if we zoom down into the endothelial wall, and at the surface of your intact endothelial wall, you have your thrombomodulin as well as your endothelial protein C receptor, EPCR. How these two receptors function to prevent clot formation is this. Now your thrombomodulin will be able to sequester and inhibit thrombins that are straying in the circulation. Alright, it will stop thrombins from going around to cleave and activate fibrinogens other than at the site of the wound itself. So anywhere else that has intact endothelial cells, if they are activated thrombin, it's going to be sequestered by your thrombomodulin. Other than your thrombomodulin, your EPCR is also there to bind to your protein C. And this protein C is especially important in your anticoagulation pathway. And this is why. Okay, so when thrombin and protein Cs are being bound together, what happens is that protein C can then be brought together into vicinity with the activated the thrombin protein. And when this happens, when they are brought together to form a complex, this allows protein C to be cleaved by thrombin, and that would then result in the activation of protein C. Okay, the formation of activated protein C or APC. So once protein C is activated by the thrombin-thrombomodulin complex, the activated protein C can then bind to another protein, another circulatory protein known as protein S. And this will allow the formation of your APC-PS complex, activated protein C and protein S complex. And this complex is essentially a protease that can cleave your clotting factors. So in this case, when APC and PS complex is formed, it can then bind and cleave your factor 5A and 7A. And that allows the degradation and inactivation of these clotting factors. So what this means is that in the blood vessels with intact endothelial cells, right, your activated protein C and protein S would then be able to remove all the clotting factors that are floating around. And this will ensure that clotting does not happen in area with intact endothelial cells, and thrombosis would not happen. Okay? So yes, if we were to revisit the coagulation cascade that you have learned from your earlier lecture, alright, you have your intrinsic pathway, your extrinsic pathway, and your common pathway, which eventually leads to the formation of your fibrin meshwork. What happens is that in areas with intact endothelial cells, there are anticoagulation factors that will serve to inhibit this coagulation cascade. And there are three main players, in which two is what you have learned. The first will be your TFPI, which inhibits your factor 7. And the second will be your APC and S system. And as what I've shared, it serves to bind and clip or degrade the factor 8, activated factor 8 and activated factor 5. With that, it shuts down the coagulation cascade from the converging part of the cascade. Now, other than TFPI and your APC and S system, there's one more main player of the anticoagulation system, and that is your antithrombin. Okay, and what is antithrombin is what we are going to learn next. So antithrombin, as what the name suggests, is particularly effective in inhibiting thrombin. How does it work? Now, you imagine that thrombin is typically trapped and localized between the fibrin mesh itself, right? Because we say that thrombin, when it's activated, is going to be restricted to the clot itself. Okay, but there will be times when there are thrombins that get leaked out and get straight into the circulation itself. And when that happens, these thrombin molecules needs to be quickly sequestered and inhibited. Otherwise, the thrombins will be floating around as starting to activate the clotting cascade, and you'll have fibrin mesh and clots forming around everywhere in the circulation, and that again is catastrophic. So to prevent this from happening, what we have here is your antithrombin, right? The antithrombin is exactly here to quickly sequester and inhibit the thrombins that are floating around in the circulation. So antithrombins are essentially plasma proteins that are being produced by the liver. They are circulatory proteins. In fact, they are actually serpins, or what we call serine protease inhibitors. What it does is that it is able to sequester those thrombins that are leaked into the circulation, and it will then be able to inhibit them and preventing them from activating the clotting cascade. Now, serpins in general are able to bind other type of serine proteases as well. So antithrombin, other than binding to thrombin, it is also able to bind and inhibit your factor X, factor 10, as well as your factor 9A. Alright, so it's not just thrombin that is inhibiting. If there's any activated factor 10 or factor 9 that are floating around, it will also be inhibited by your antithrombin too. So with this, you can see that clearly there are multiple mechanisms to ensure that clotting is really localized only to the damaged vessel, and anything that leaks out will be quickly sequestered and inhibited so that thrombosis does not happen. With that, this comes to a quick summary of the three main players of our anticoagulation system. You have your TFPI, your APC and S complex, and finally your antithrombin, which serve to inhibit not just your thrombin, but also your activated factor 10 and your activated factor 9 as well. Okay, I know it's content heavy. We're going to have a quick stretch break, but we are not going to have the next part recorded as a separate recording, because the next part is going to be rather short, so we are going to push through it. So take a quick sip of water, refresh your mind, and we are going to get started. Okay, so we have learned about plaque and clot prevention, and we said that plaque prevention is executed by your nitric oxide and prostacyclins released by the intact endothelial cells, whereas clot for prevention is executed by your APC and S, your antithrombin, and your TFPI. So we are going to move next to the clot removal, or your fibrinolysis. And when it comes to clot removal, it needs to only happen after the wound has fully healed. Because if clot removal is allowed at a site in which the wound has not fully healed, then the clot can be removed and bleeding can happen when the wound is still fresh. So we do not want that to happen. And the first step towards the removal of your clot is actually the degradation of your fibrin mesh, because that's when the insoluble mesh is taken away and the clot can be dissolved. And for this to happen, you need a protease, and that protease is your plasmid. Okay, plasmid is that main protease that mediates fibrinolysis. Now, as of all proteases, it needs to be regulated. And how is plasmid regulated? Essentially, plasmids are always present as an inactivated enzyme, and that is your plasminogen. Alright, the zymogen of plasmid is known as plasminogen. So plasminogens are circulating proteins that are freely circulating around in the plasma. Now, it can only be activated into plasmid in the presence of another activator known as your tissue plasminogen activator. Alright, and your TPA here is only released by your intact endothelial cells. So what this means is that if the wound is still fresh, the endothelial cells are damaged, you will not have TPA being removed and you will not have plasmid being activated. Moreover, your TPA can only activate your plasmid in the presence of fibrin. Okay, so TPA can't go around in the circulation activating your plasminogen if there's no fibrin molecules. And after plasminogen is being activated into plasmid, that is when it can start chewing up your fibrin mesh, and all the fibrin degradation products will be released into the circulation. And that's how clot removal happens. So then the critical question is here. How does the body ensure that plasmid only activates, is only activated and start degrading your fibrin mesh at the repaired wound itself? Alright, how can you make sure that fibrinolysis is only localized to the wound that has been fully repaired and not anywhere else? And the body does this through 3 mechanisms. Okay, first here, fibrinolysis is restricted to the right place. Number 1, through the absence of TPA from damaged vessels. So as what I've shared before, TPA is only released by your intact endothelial cells. So when the wound is still damaged, the endothelial cells that are damaged would not release TPA, and hence you won't have TPA floating around in the vicinity of a fresh wound. The second mechanisms in which plasmid is restricted only to the repaired wound is the fact that TPA can only activate plasminogen in the presence of fibrin. So what this means is that if you have TPA floating around, alright, and if it meets plasminogen here without the presence of fibrin, it would not be able to activate the plasmid here, and hence you won't be having plasmid floating around trying to activate or trying to cleave all the clots even when the wound is not fully healed. Now the third factor, which is the most important factor here, are actually the presence of circulatory PAI and antiplasmid. PAI here stands for plasmid activator inhibitor. Essentially is your TPA inhibitors. Alright, so to illustrate this better, now you can imagine that sometimes you may have plasmids, activated plasmids, and also TPA being straight off into the circulation. So occasionally there will be plasmid and TPA floating around. And to make sure that these stray plasmids doesn't go around cleaving off fibrins when it is not needed yet, there are other circulatory proteins that helps to quickly scavenge off all these stray plasmids, and that would be your antiplasmids as well as your PAI. So your TPA and plasmid that strays into the circulation will be rapidly sequestered and inhibited by your PAI and your antiplasmid. With that, what this means is that your plasmid activation and fibrinolysis will only be restricted to the clots with wounds that are fully recovered. Okay, so this pretty much sums up the regulation of fibrinolysis. But I want to take this opportunity to also highlight another factor here that is critical for your learning, and that is your fibrin degradation product. Now you realize that I don't usually highlight the degradation products from the protease, but the NDPs here as especially important because they actually have some clinical value in fact, it's quite a bit of clinical value. And why so is something that we will learn next. Alright, so what are fibrin degradation products? Now, if you look at your fibrin strands, alright, and if we zoom into the fibrin monomers, you realize that the fibrin monomers are actually connected or cross-linked to each other through this particular part of the fibrin monomers called the D domain. Okay, so each fibrin monomers has D domains that are connected to each other. And these D domains, when it's cleaved by your plasmid, or your fibrin strands when it's cleaved by your plasmid, it's actually cleaved in a very standard manner, in such a way that when fibrin strands are degraded, the D domains are being released together with its cross-link. And these D domains that are linked are known as your D dimers. Alright, so your fibrin degradation products will have all the D dimers in them. And why are these D dimers important? Right, so these D dimers are essentially fragments of the fibrils that contains the two D domains as well as the cross-link that link it together. And D dimers are important because it actually serves as an indicator of a thrombotic event. What this means is this. Now, if we were to measure the circulatory D dimer in the serum, there's always a standard or normal range that we usually see in healthy adults. But what happens is that when we see elevated D dimers in the serum, and that person doesn't have huge clots from injuries or fresh wound, what this means is that there can potentially be thrombotic event or thrombosis happening in somewhere else in the body. Alright, so D dimer is a good indicator for us to have a sense of whether thrombosis is happening in the body. How do we apply this? It's something that we will learn in the in-person lecture later on. Okay, so this pretty much sums up the recorded session. What we have learned today, firstly, will be the fact that your appropriate and timely clot really needs a tight regulation of your clot formation as well as your clot inhibition. So that tight balance is very important. The second important key message is that your intact vascular endothelial lining is absolutely critical to ensure that the coagulation system functions optimally. The third and final learning from this recording is the fact that your D dimer level in the serum is a good indicator of thrombotic events. How do we apply this is something that we will try and do during the in-person lecture itself. So please attend the in-person lecture, and I'll see you then. Thank you.